So our next speaker is Fred Gmitter from the Citrus Research and Education Center at the University of Florida. Fred's a leader in the development of genetic and genomic methods in citrus improvement. On the practical side, he's been involved in the generation of several new cultivars. There's a really great fruit market in Berkeley, and I go there, and the whole entryway is all filled with different citrus crops. And usually after I go there, I call Fred up and I tell him something exciting that I found, and he tells me, why I know nothing about citrus. Fred's a leader in developing efficient ways to work with improved plants, to clone and manipulate citrus genes. You might say his specialty is squeezing the most out of the citrus germplasm. I'm sure you've heard those before. Citrus beating with a twist. For these achievements, he's recently been named the Florida Researcher of the Year by the Agricultural Industry Association there. For the past 5 years, although I'm sure it seems like a lot more, Fred's been the leader of the International Citrus Genome Consortium, which involves a whole host of countries, the US, Spain, Brazil, Italy, France, and several others I've forgotten. And Fred's really been driving this group to produce not just one, but several citrus genomes. And we're still in the process of analyzing them, but I think there's some interesting stories there. Fred's been on the front lines fighting the citrus disease greening, which is a potentially very devastating disease of citrus crops. And he's going to tell us today about how genome-based science is shaping the future of the world's citrus industries.
Thanks, Dan. You gave half of my talk already. First of all, I just want to express my gratitude for the opportunity, the privilege to participate in a meeting like this. And in kind of echoing a theme that we heard in the talk yesterday, it's important that I point out to you that I also have my undergraduate degree in English, as our speaker did yesterday in the evening, and I also dabble some in genomic science, as I think he does. And also in keeping with the theme presented in the talk yesterday, I should be able to tell you what I do and who I am in 2 brief sentences. I am a citrus breeder geneticist. My goal in life is to develop new varieties of citrus that are better tasting, that grow better, that are resistant to disease, and that will help to drive the economic engine that our citrus industries are in the world. And I'll move on from this point. Some citrus facts. Citrus, you may or may not know, is actually the #1 fruit crop grown in the world. Not only in the world, but in the United States. Some might think bananas or apples or grapes, but in fact, if you look at the FAO statistics, you'll find citrus is #1. The major producing regions of the world are in Brazil, in the US, in China, which now has more citrus trees in the ground than any other country on Earth, and the Mediterranean basin area. Florida is very important in the U.S. citrus industry because it produces 75% of the U.S. production of citrus. The vast majority of that is turned into juice, squeezed, if you will, Dan. The value of that industry in Florida to the farmers is $1 billion US. And if you look at the total impact across the economy in the state of Florida, it's worth $10 billion. Sweet orange juice is a global commodity, like oil, like cotton, like wheat, like soybeans, and it's traded as such. Aside from that, the rest of the world is very much focused on citrus as a fresh fruit industry. Aside from being a global commodity, it's also widely grown throughout underdeveloped and developing areas of the world where it provides an important source of nutrients, particularly vitamin C. Some of the biological facts: citrus is a cross-pollinated diploid, a fairly small genome, estimated previously to be about 360 million bases. The results of genome sequencing have shown that those early estimates are a little bit on the high side. The genus is full of examples of cross and self-incompatibility. It's a very difficult plant to breed. Aside from the incompatibilities, there's abundant male and female sterility that can be found. The plants are large, So if you're running a breeding program and planting out 10,000 seedlings from a cross, you're looking at tremendous investments in land area, in care and management, and these are large-sized plants. So it's not like working with Arabidopsis at all. There's widespread apomixis through a phenomenon known as nucellar embryony. The nucellus is a maternal tissue in the seed, and nucellar tissue generates somatic embryos. So if you're a breeder and you're making crosses and you grow out 1,000 seedlings, depending on the kind of citrus you started with, you may simply be cloning the maternal genotype and not finding any hybrids, no recombinants, nothing at all. There's a tremendous genetic diversity within the genus and in some sexually related genera. So the introgression of genes, traits, and characteristics is possible. But within the cultivated species, the genetic diversity is extremely narrow. We talk about species in citrus, and we talk about Citrus sinensis, the sweet orange, or Citrus paradisi, the grapefruit. The reality is these are not biological species. And in the case of grapefruit, we have some good historical documentation of its origin and evolution. The first grapefruit originated in the New World probably around 1700 in Barbados, most likely. Earlier times, the European explorers were traveling around the world and moving plants and animals and all sorts of things. They knew if they planted an orange seed, they would recover an orange tree because of the nucellar embryony. If they planted a lemon seed, they would get a lemon tree, and so on. They were kind of blown away when they went to Southeast Asia and found these large fruited citrus that we call pumelo, Citrus maxima, or Citrus grandis. So they brought seeds of those to the New World from Southeast Asia, planted them. But pumelo is different from many other kinds of citrus in that it produces only zygotic offspring. So when they planted these pumelo seedlings, they got these hybrid swarms. And one of the very first ones that was recognized was the very first original primeval grapefruit tree. There was a French count. Some say he was a pirate. Probably both are true. His name was Philippi. And he brought some grapefruit trees into Safety Harbor, Florida. This is in Tampa Bay in 1823. He was really fond of his grapefruit trees, and he gave seeds and plants to all his friends and relatives. And they began to grow grapefruit. It was a white-fleshed variety. It had 50 to 60 seeds per fruit. A few decades went by, and some folks found the so-called Marsh grapefruit, which also is white, but the seeds were gone. So people thought this was a nice thing. The seeds are gone. Let's grow this one. And it was propagated and spread rapidly. In a few more years, a bud sport mutation, a limb on a tree, a white-fleshed tree, was found that had fruit. When you cut it, was pink inside. Everybody thought that was really, really good. So they propagated more of that, and things went on, and eventually over time they found pigmentation on the peel and darker pigmentation. So what we have here is not a species, but really the diversification from one initial source. If we look at the story of sweet orange, essentially it's the same. The sweet orange perhaps originated 1,000 years BC in China, maybe longer than that. So we don't have a very good and clear historical record of the evolution of orange, but as we look at all the different kinds of orange that exist in the world today, it's a very similar story. No oranges and no grapefruit have ever been developed by making crosses, by sexual hybridization and breeding, which is a really depressing thing for me to realize when I was hired as a citrus breeder. So we have a number of objectives in our breeding program. Among the most important of these is disease resistance, but if you look at any plant breeding program, you also find improvements in quality, improvements in yield, and citrus, being a complex plant, is grown as a scion grafted to a rootstock, and there are even more characteristics for rootstock improvement that we're very much interested in. But being a plant that's not very amenable to genetic studies, having a 4 to 6 year juvenile period and the large size and so on, there's very, very limited information about the genetic control of any of these important traits. This is the first impact the first involvement of genomics and the value of genome sequencing to improve our ability to improve these plants. I have to introduce this disease that Dan referred to as citrus greening. Its official name is Huanglongbing, and depending on which part of China you're from, it either means yellow dragon disease or yellow shoot disease. It's a nasty, nasty disease. It's insect vectored. It's a phloem-limited fastidious proteobacterium. It has a latency period in plants anywhere from 3 months to 3 years, actually. And while it's living in the plants and the plants are asymptomatic, the insect vector is feeding on the plants and moving this disease everywhere. And when it finally is expressed in the citrus groves, it's too late to do anything about it. The pathologist's point of view is that we should identify the infected trees, remove them as quickly as possible to reduce inoculum, control the vector, and everything will be fine. That's good if you're a pathologist, but if you're a citrus grower and you find a tree with one branch that has some symptoms on it and the rest of the tree is full of fruit, it's awfully hard to justify removing those trees. And very quickly the citrus growers saw that they could be out of business. So it's a frightening thing. There's no known genetic resistance within the citrus germplasm pool. There are different degrees of tolerance. Plants such as lemon, for example, can become infected, look very symptomatic in the autumn of the year, and then produce a new flush in the subsequent spring and just keep going. They kind of outgrow the disease, whereas sweet oranges, when they become infected, generally decline quickly and die, unless nutrient solutions are applied to foliage to try and circumvent the decline of the tree. This was first found in Asia in 1900. In 2004, it was found to be widespread in Brazil. In 2005, we also woke up and found it to be very widespread in Florida. And if you talk to anyone in the world of citrus, they'll tell you that Huanglongbing is the greatest threat to the survival of the industry. As with many disasters, sometimes good things happen because this motivated many of the industries in the world to generate funds for research. And one of the first things they were convinced was a worthwhile investment for their funding was to move forward with some citrus genome sequencing projects. This is what the early symptoms of the disease look like. It's characterized by chlorosis of the leaves. With some fruit varieties, you can see what's called Hongbitze in Chinese. It means red nose. It's a very typical characteristic, even when the leaves are asymptomatic. It's a disaster, and it's not my visiting this place that's the disaster. It's the citrus trees that you see there in a very serious state of decline. It's rather difficult to see him, but there is the insect vector called the Asian citrus psyllid, a phloem-feeding insect. In the foreground is an orchard of Ponkan mandarin in Guangdong province that's completely devastated. If you look in the background, though, there's some healthy citrus orchards growing right here. Their life expectancy is very, very short. So the economics of this disease are really critical. In 2003, we established the International Citrus Genome Consortium, and we had a list of goals, as most of the other genome consortiums have, particularly with our interest in whole genome sequences. At that point in time, we were looking at an estimated cost of $10 million to sequence a genome. It's amazing how things have changed. We began our quest as a citrus community by focusing on sweet orange, because sweet orange is, in fact, the most widely grown kind of citrus in the world. In meetings with folks here at JGI and learning about the experience with poplar, we were informed that if there was a haploid of citrus, it would make life a whole lot easier for everyone involved in our project. And so we did, in fact, identify some haploids and selected one from a clementine mandarin for the sequencing project. This was done with Sanger-based sequencing. I think we were the last, or very near to the last, of the Sanger projects done at JGI. In parallel, we also began a project sequencing orange in collaboration with 454 and with JGI. So the clementine partners are listed here from Genoscope, from IGA in Italy, the good folks at JGI, Hudson Alpha, and a small company in Spain called Life Sequencing that did some 454 sequencing for us. The citrus partners are listed here. Primarily these nations, Brazil, France, Italy, Spain, and US, were the ones that contributed financially to this project. And when I talk with growers, I talk about the promise of genomics, and the most critical point for us and for the growers to understand is all of the omics that we talk about must essentially be tied to plant performance, to aspects of plant performance, to have any real value and meaning. And sometimes that's a difficult thing to communicate to growers. We have a version 0.9 that's on phytosome of the clementine genome. It's a pretty good assembly. The N and L50 figures are here. The genome size, we estimate it to be, or what's been sequenced, is about 296 million bases. We've got a new version that's not yet out, although it has been shared with some in the community with even better scaffold N and L50 figures. The Sweet Orange project was a collaboration, as I mentioned, of my lab and other folks at the University of Florida, primarily with Dan and Simon here. The folks at Roche 454 contributed tremendously, as did Mark Borodovsky from Georgia Tech. This is a much more fragmented genome, but if we compare the numbers of predicted genes in this and clementine, we find that they're essentially identical. So though it's fragmented, we've accessed most of the gene space within this genome. And what we've been doing now in recent times has been to compare the clementine and the sweet orange to try to have an understanding of the origin of sweet orange. I mentioned that it's not a true species. There are two true species that seem to be involved in its phylogeny, the mandarin and the pomelo. And by looking at differences across the haplotypes of the 9 basic chromosomes, we can assign certain regions of the haplotype as being mandarin, mandarin pomelo, or pomelo pomelo. This is important for a citrus breeder's dream of recreating the sweet orange, but modifying certain critical characteristics, perhaps identifying genes for disease tolerance that could be incorporated as we rebuild the sweet orange. This would tell us the kinds of segments, chromosomal segments, we need to select for. But I want to come back to HLB. Again, here's another photo of how horrible the trees look, how horrible the fruit is, and very typical asymmetric fruit. HLB is a carbohydrate problem in plants. There's a plugging of the phloem tissue and an accumulation of starch in the above-ground parts of the plant, which results in a drastic reduction in starch reserves in the roots. This is a photograph, a micrograph. This is a healthy sweet orange leaf petiole. You can see xylem, normal, healthy phloem. This is a petiole of an asymptomatic leaf, which is well on its way to becoming symptomatic. You see collapse of the phloem tissue. You begin to see plugging of the vessels with callose, accumulation of starch, and this is from a symptomatic leaf where you can see an awful lot of starch accumulating and callose. So essentially, the plant loses its ability to move photosynthate through the phloem to the roots, and everything falls apart from that point in time. So we're interested in trying to dissect the mechanisms of disease, to compare infected and healthy, to look at tolerant versus susceptible. Again, the example of lemons and oranges, one which outgrows the disease and one which doesn't. There's interest in looking at the impacts of these different nutritional regimes. I won't talk about that, nor about rootstocks today, but those are other areas that are vigorously being researched. This panel up on top is a kind of lemon called rough lemon, and this is sweet orange. And what you see here are water controls. These are healthy leaves that have been abraded in a fluorescent compound that is indicative of phloem loading, and you can see here these veins in sweet orange and in lemon. Clearly the material's moving through. In rough lemon, this is an asymptomatic leaf, and you can still see evidence of phloem loading, but when you look at the same thing in sweet orange, you see it's drastically reduced. And here in the fourth line are symptomatic leaves. And again, you still see some phloem activity in the rough lemon and absolutely nothing in the sweet orange. So there's something going on with the phloem and its function. So we and the citrus community in general is taking a holistic approach looking at transcriptomics, both microarray and RNA-seq projects, which ultimately are dependent for their best interpretation on good genome sequences, proteomics, metabolomics, and so on. This is a heat diagram of a microarray experiment. This is looking at rough lemon. What's critical to notice here is that several genes involved in cell wall biosynthesis are upregulated. Very few of these, looking at the wrong thing, very few PR-related genes are upregulated. When we look at the same thing with sweet orange, we find cell wall genes are severely downregulated. You find a whole lot of upregulation of MIB transcription factors, PR proteins. So not only do you have this collapse of the phloem in sweet orange, but you have a late-stage initiation of defense mechanisms, overexpression of defense mechanisms that essentially uses up all of the reserves of the sweet orange plant. We don't see anything like that in the lemon. So a lot more genes with altered expression in orange than lemon. These cell wall associated genes upregulated in lemon but downregulated in orange. And this little business here about less phloem regeneration and even loading in the infected rough lemon. So this seems to be some sort of mechanism. What next? We view the genome sequences, and there are many sequences coming. Currently, there are at least 5 genome sequences available in citrus, and we understand that a colleague in Spain has a project where already 180 citrus genomes have been sequenced. So there's an awful lot going on in our world. As a breeder, one of the more interesting things is to use this resource to develop good marker-assisted breeding technology. But from a more fundamental point of view, understanding host pathogen interactions is critical, particularly as we look at this disease, Huanglongbing. Taking best guesses to unambiguity, the best guesses being what we see when we look at microarray studies, to have a full understanding of the full gene sequence of what seems to be affected as we look at the results from microarrays, and to identify the upstream and downstream elements that are involved in these processes as well. Answer that question, we're trying. Aside from all the disease and the fundamental research on this particular disease, there's work going on to elucidate metabolic pathways, particularly the carotenoids, which are one of the favorites in the citrus world. Everyone likes orange and deep orange and blood orange and so on. So there's applications of genomics to this. There are these chemicals called furanocoumarins that are implicated in the grapefruit juice effect. And there's a big interest in trying to understand the control of that pathway and to be able to manipulate that. And of course, everyone always is looking for targeted disease resistance. This is a graph of drugs taken with grapefruit juice and drugs taken without grapefruit juice, of the level, the concentration of medications in the human bloodstream. Grapefruit inhibits a cytochrome P450 in the human liver, which then doesn't metabolize drugs that are consumed at the rate that it should. And these drugs then enter the bloodstream at very much higher levels and lead potentially to very dangerous side effects. This is a real critical issue with grapefruit, because grapefruit consumption in the world has gone down mainly because of this. The market for grapefruit is generally old people. The market and the market for Lipitor, Crestor, and blood pressure medications are generally old people. And so we've seen this market really collapse and prices for growers really go to hell. We've made some crosses over the years of some pomelos with grapefruit, mainly to improve fruit color. But when we evaluated some of these things for the furanocoumarins, we found something interesting. This is Hudson grapefruit, and it shows 3 of 4 of the family of furanocoumarins, and these are among the most active, metabolically active furanocoumarins. And contrast that with the pomelo that we used as a seed parent, which has a high concentration of imperatorin, which is an inactive furanocoumarin that also has some health beneficial properties. When we crossed these things, we saw that we got 2 kinds of offspring. We got those that resembled the safe-to-eat pomelo and those that resembled and maybe were even worse than the not-so-safe-to-eat grapefruit. We've done digital gene expression looking at all the genes that we're aware of in this pathway. We found that these 3 bad boys were correlated. And so there's an upstream, we hypothesize, an upstream regulatory sequence that controls this and appears to be inherited as a single gene, a single Mendelian gene. So we'd like to understand what that is, and using the genome sequences, we hope to be able to learn more about that and to be able to use that perhaps in a transgenic strategy on ordinary grapefruit or in a marker-assisted breeding strategy to be able to make selections at a very early stage. Right now, we have a variety that's been approved for release. This is not a red grapefruit. This is a hybrid of a pomelo with a tetraploid red grapefruit. It has virtually no activity, no enzyme inhibitive activity, so it should be safe to eat. It happens to be a triploid, so it's seedless as well. And it's a little bit sweeter and a little bit less sour and a little bit less bitter than grapefruit. So this is something that we got lucky and selected, but we'd like, in fact, we have hundreds and hundreds more hybrids from these kinds of families, and we'd like to be able to select when the plant is this big for this characteristic, as opposed to growing out acres and acres of them and waiting 6 to 7 years to identify the low furanocoumarin types. I get involved with all sorts of things besides genomics and breeding. One of the things I'm involved with is trying to understand what it is that drives the market, what drives consumers to buy and to buy again citrus products. And one of the more interesting things we've found is that, and these are from mall intercept studies, is that generally American consumers are more focused on freshness and flavor, and freshness really is another aspect of flavor. That's one of the most important things. And secondly here is appearance, and that generally translates to the color of the fruit. So color and flavor are very important things. And so going from this, we've initiated studies to look, in this case, at the content of aromatic volatiles and their impacts on flavor, and to try and identify a genetic basis and association basis for different volatiles and different profiles that consumers find desirable. And ultimately, as a breeder, I want to get to the underlying genetics of these characteristics, of the specific points within them, and to be able to make selections a more efficient process. Taking the big view now, this is something that was in Nature in 2011. It's 4 decades of genomic research and five domains of genomic research, if you will. This is pre-human genome, and as you look over time, the applications where we're actually having impact of genomic science on human health is probably 10, 15, 20, 25, 30 years down the road. When I show this to citrus growers, they're really angry with me because they thought that if they had the genome sequence, their Huanglongbing problem would be solved. But it's important to keep this perspective because it's not just another genome. It's more than that. And this is what Mark Poznansky was quoted as saying, is that genomics will be to the future what broadband has been to communication and what highways have been to transportation. And so that's the message I leave with my citrus growers, that we haven't solved your problem, but we've developed an extremely powerful new way to study the problem and hopefully get to solutions in the near future. The folks who supported this work, the international project, funds came primarily from Brazil, France, Italy, and Spain, and the US. And the folks in the US who contributed were the Florida citrus growers, the first to put money on the table for the international sequencing project. Roche 454 Life Sciences has been a huge contributor, and certainly JGI has been with us all the way, and some support from within my university. And lastly, to prove that I am a citrus breeder, this is fruit of the very first variety that we released from our breeding program. This is fruit called Sugar Bell. And so if you ever see Sugar Bell, and there has been some marketed in Berkeley, look for it. It's a good variety. So thanks.
Thank you, Fred. Any questions? Jim?
So how close are lemon and sweet orange, and are there potential breeding strategies to try and understand the resistance that appears to occur in lemon and not sweet orange?
They're not so close, but there are. Citrus is a very incestuous genus, and you can ask Albert. He's been looking real closely at this. Nothing is pure. There's an awful lot of movement of parts of the genome from one to another. What we're looking at in terms of exploiting this tolerance that we find from lemon, we've identified actually 6 or 7 genes that appear to be very critical, central to this process. And we're working with some folks in Argentina and in Spain now developing silencing constructs, essentially, that we can use in some long-term transient gene expression assays. That's an oxymoron, I recognize, but there's some little tricks that people have been playing, and we can silence genes for up to 18 months in citrus plants in a transient fashion. And to put these into plants and then to test what impact they have, if any, on the expression of disease from this pathogen. Actually, it's kind of a very quick way to look at more than just these 5 or 6 that we think are most interesting. I mean, if we really show you our list, it's about 30 or 40 genes, and that's just not a practical thing we can approach.
Hi, is there any chance that you could get the aphid to introduce a virus? The enemy of my enemy is my friend?
Almost anything anyone here can imagine is probably already being researched because of the millions of dollars that have been generated by the industry in response to this fear factor, if you will. And that's one of the strategies that's being looked at. There's a virus called citrus Tristeza virus, which also happens to be a phloem-limited pathogen, just like Huanglongbing. And some pathologists have engineered Tristeza virus, CTV, with antimicrobial peptides. And the idea is, yeah, you could do it by aphid transmission, or you could very simply just graft, an a CTV-infected piece on the tree. So people are looking at that, and that may in fact be a strategy.
Okay, if there are no further questions for Fred, let's thank him again.
Thank you.